Hi everyone, thank you for joining the fourth webinar in our series, Use of New Approach Methodologies for the Risk Assessment of Pesticides. This series is being co-organized by PETA India, the Society for Alternatives to Animal Experiments in India, and the Biological Agri Solutions Association of India. I'm Dr. Ankita Pandey and I'm an advisor to PETA India. I'll be co-moderating today's webinar with Dr. Akbar Shah from the Society for Alternatives to Animal Experiments India. Today, we will hear first from Dr. Parvathi MVS, Aurigene Pharmaceutical Services Limited, who will be presenting on use of in vitro and ex vivo eye irritation corrosion test methods for the toxicity assessment of pesticides. Before I introduce the first speaker, I would like to point out that the information about the webinar series can be found at the link on this slide. There will be time for questions for the speakers at the end of the presentations. Everyone is on mute, but you can type questions or comments in the question section within your GoToWebinar toolbar at any time during the presentations. This toolbar should be on the right-hand side of the screen. Please do mention to which speaker the question is being addressed to. Speaking first today will be Dr. Parvathi MVS. Dr. Parvathi is a highly accomplished professional in the field of toxicology with a specialization in vitro and genetic toxicology. She holds a doctorate in molecular oncology toxicology from the University of Madras. She is a European registered toxicologist and has accumulated over 16 years of industry experience. Dr. Parvathi began her career in 2007 at IBAT, a GLP CRO wherein she actively participated in safety testing of agrochemicals, cosmetic ingredients, dyes, to name a few. Later, she joined Aurigene Life Sciences and then Vimta Labs. Currently, Dr. Parvathi is associated with Aurigene Pharmaceutical Services Limited, where she specializes in chemical safety testing. Dr. Parvathi has also actively engaged in professional organizations, including being a member of the Society of Toxicology in the USA. It is now my pleasure to turn the floor over to Dr. Parvathi.
Good evening, everyone. So today I will be talking about the ocular toxicity predicting in vitro and ex vivo models. Used for the classification of agrochemicals in this session. So briefly to brush upon on the eye exposure to agrochemicals. Generally, eye damage occurs by accidental splashing or exposure to chemical particles, vapors, or gases. The substances contacting the surface of the human eye naturally, they expel in less than 2 minutes. That is, almost 80% of the chemical would be washed off with the tears. So short-term exposure assays were developed with this concept to understand the toxicological effects of exposed chemicals. So traditionally used animal model was ocular toxicity evaluation using in vivo rabbit race test with the OECD test guideline 405, where the rabbit eye is exposed to chemical and the degree of eye irritation or serious eye damage is evaluated by scoring lesions of conjunctiva, cornea, and iris at specific intervals. And if we look into the stats, the US EPA receives almost more than 200 rabbit eye irritation tests each year for the data on agrochemicals. So we see that a need for 3 R's, that is the refinement, replacement, and reduction in animal model testing is identified and mandated by the agencies these days, which led to the use of alternatives to animal testing. And coming to the classification of eye damage, there are 3 classes which are categorized by UNGHS, Globally Harmonized System, wherein category 1 accounts to the serious eye damage, which is irreversible effects on the eye irritation within 21 period of within the observation of 21 days in rabbit race eye test. And the same classification goes with US EPA as category 1. Then the common category of category 2 refers to the eye irritation, wherein category 2A is referred to or classified as eye irritation where reversible effects on the eye which are fully reversible with the effects within observed period of 21 days, and the category 2 accounts with US EPA category. Category 2B of UNGHS accounts to mild irritation where the reversible effects on the eye are observed within a period of 7 days, fully reversible, and no category where the chemical accounts to non-corrosive, non-irritant and not classified. When it comes to the US EPA category, 3 and 4 both are considered to be mild to non-corrosive. So this is the eye damage classification. There comes the eye damage and irritation predicting methods, where various validated in vitro and ex vivo alternative methods are available to test for the ocular toxicity. And these are listed out in this slide based on their ability to detect the severity of damage to the cornea. You can see the picture where the corneal epithelium, stroma and endothelium are placed, and the epithelium is on the superficial layer. The squamous epithelium is the outer layer of the cornea. So assays are designed based on the severity where they cause the damage to the parts of the cornea. Here comes the organotypic ex vivo assays, which are listed here, like bovine corneal opacity LLBO test, isolated chicken eye test, isolated rabbit eye test, porcine cornea opacity reversibility assays, ex vivo eye irritation test, which are used to check and detect the damage at the corneal epithelial, stroma and endothelium level. Then comes the reconstituted three-dimensional human tissue assays, which targets the epithelium, and they are reconstructed human cornea-like epithelium RHE test, epiocular time to toxicity ET50 determining assay using our HCE model, lab side cornea model 24, which is a Me2 assay, and vitreous eye irritancy test. These are mostly the validated assays which are placed into the guidelines. Then coming to the third list of assays, those are cytotoxic and cell function based assays, in vitro assays, which are used to detect the damage at the outer lining of the cornea. They are listed as short time exposure in vitro test, fluorescein leakage test, neutral red release test, cytosensor microphysiometer test. Additionally, few in chemico assays are in place to understand the epithelial damage to cornea, like ocular irritation method and OptiSafe test. When we go in brief with reconstructed 3D human tissue assays, we have a preferred list of assays which include RHCE, lab side cornea model, and epiocular time to toxicity method. And other RHCE models are also placed into the guideline 492 of OECD, which holds equal validity. If we see the RHCE test, the basic test and commonly used test, the validity is based on OECD 492 guideline, and the test system used is reconstructed non-keratinized multi-layered human corneal epithelium, and the exposure for neat liquids would be 1 to 30 minutes, and for neat solids it is 3 to 24 hours, 3 to 24 hours. And the endpoint measured is cytotoxicity with MTT reduction assay, and the applicability is is used to detect the UNGHS non-category of chemicals, including all agrochemical formulations. Then coming to the epiocular time to toxicity assay, the assay goes same as OECD 492 RHCE method, and it is validated as 492B. And the same test system of human non-keratinized multi-layer 3D tissue inserts will be used. And the exposure for neat liquids would be 5 minutes, and 16 minutes for 20% formulation, and 120 minutes for 20% formulation again. When it comes to solids, the neat solids will be exposed to 30 minutes and 120 minutes. The outcome would be the measurement of cytotoxicity using MTT assay, and it is again used to classify the chemicals as category 1, 2 and non-category. The other assay which is validated is the lab side cornea model 24 eye irritation test, which is a part of OECD 492. It is optimized and the test system is a RHCE 3D tissue insert. The exposure would be for 1 minute for liquids and 24 hours for solids. Endpoint is based on MTT based reduction and WST or WST, that is water soluble tetrazolium dye 8 or 1 based assays. And the applicability of this tissue model would be for classifying the chemicals to 1, 2 and non-category. So here is the image where the flow chart is represented, that the tissues will be treated with the chemical and they will be rinsed after incubation period and post incubation followed by MTT reduction and isopropanol extraction and reading of the plates at defined nanometers of 570 or 450 or so on. Looking at the classification based on endpoint from RHCE models, the classification goes like, when it comes to the no category classification, the mean tissue viability with RHCE model should be greater than 60 for categorizing the chemicals as no category. And if it is less than 60%, no prediction can be made. And the same applies with skin ethic model for liquids protocol. And for the skin ethic model for solids, the mean viability should be greater than 50%. And for lab side cornea model, it is greater than 40% and less than for no prediction. And with MCTT HCE for liquids and solids, which is an additional model referred in the guideline, the mean viability for liquids should be greater than 35, and for solids, it is greater than 60% viability required to categorize the chemicals with their outcome. And when we look into the epiocular TTT models, the classification goes with mean tissue viability, which is greater than 50 with all time treatments. It is a no category chemical. And if it is less than 50, then it is categorized as category 1. And any other combination of values shall be put into category 2. The same applies with HCE TTS for solids, wherein this viability should be greater than 40% after 30 minutes and 60% after 120 minutes. And the other assay is the vitreous eye irritation test, which we have listed here. The validity is based on OECD 494 validation and putting into the guideline. The test system is again reconstructed non-keratinized multi-layered human corneal epithelium. And the exposure goes with 3 minutes at 2.5% formulation of the chemical. Endpoint would be the damage to the epithelial barrier function measured by time-dependent changes in transepithelial electrical resistance, that is the TR measurement. This method is used to detect UNGHS non-category chemicals. And this is the pictorial representation of the chemical where the electrode is placed onto the tissue inserts and the tear recorder will record the tear measurement and the prediction will be made. Then coming to the major ex vivo assays where models are developed which are close to direct organ exposure. So the main assays listed are bovine corneal opacity, LLBO, that is laser light based opacity measurement, and isolated chicken eye test. Both these tests are validated with OECD test guideline number 437 and 438. Coming to the BCOP LLOB method, it uses cornea isolated from bovine eyes obtained from animals at slaughterhouses. Exposure is for 10 minutes for neat liquids, and surfactants, it is 10% wide by volume formulation. And the 4 hours exposure goes with non-surfactant solids. Endpoint is the measurement of corneal opacity, loss of barrier function, that is permeability, using fluorescein dye. This method is used to categorize chemicals to category 1. All type of chemicals are tested using this method. And this is the representation of the method, pictorial representation over here, where the corneal bovine cornea is separated and placed onto the chamber, and test item would be injected into the chamber, and then after incubation, the opacity measurement will be measured using conventional opacity meter or the laser light based opacitometer. And when it comes to the classification criteria, the in vitro irritation score less than 3 accounts to no category with conventional models, and with LLBO, the irritation score should be less than 30. And in vitro score of greater than 55, and in vitro score with LLBO with greater than 30 and less than 145 of lux, and OD of greater than 2.5 accounts to category 1. And the range in between 3 to 55 and 30 to 145 and OD 2.5, no standalone prediction can be made with this assay. Then coming to the next assay in this genre are the isolated chicken eye test, which is again validated as guideline number 438. It uses chicken eyes isolated from abattoir animals, and all chemicals can be tested neat for 10 minutes. And the corneal opacity is the endpoint, along with fluorescein retention, corneal swelling, morphological damage, and histology. Application is for categorization of category 1 chemicals, and all type of chemicals can be tested. This is a pictorial representation of chicken eye test. And coming to the cytotoxicity and cell function based in vitro assays, simple and robust cytotoxicity and cell function based assays like fluorescein leakage test and short time exposure tests are available for classification and labeling of chemicals. Both these assays are validated. Fluorescein leakage test accounts to the guideline numbers 460 of OECD. Test system used is confluent monolayer of MDCK CB997 tubular epithelial cell line. These cell line are exposed to series of 5 concentration of chemicals for 1 minute each. Endpoint would be transepithelial permeability to fluorescence. And application is that it is used for UNGHS category only to the soluble chemicals and those that form a stable suspension during testing. This is a specification of fluorescein leakage test. So this is a pictorial representation of how the test is carried out, where the fluorescein leakage is measured. There comes short time exposure test. Here, the assay goes with OECD test guideline 491. And the test system here is confluent monolayer of SIRC stratum rabbit cell line and rabbit eye cornea cell line. And the exposure will be for 5 minutes at 5% and 0.05% of the formulation. Then coming to the endpoint, it is a simple MTT-based cytotoxicity measurement. And this method is used to classify the chemicals under category 1, that is for serious eye damage, only to the soluble chemicals and those that form a soluble suspension during testing. So just a pictorial representation of the cell line based assays here. There comes the additional tests that are in chemical models, that is as mentioned. These macromolecular matrix assays are made available to support the classification of chemicals and labeling. These assays are in vitro macromolecular, ocular irritation methods, and OptiSafe method. Both are similar though. And this method is validated and put up into OECD as 496. Test system is the transparent macromolecular matrix, a compound mixture of proteins, glycoproteins, carbohydrates, lipids, and low molecular weight components with that mimics the highly ordered structure of cornea in chemical. And exposure would be made with 5 different concentrations of the test chemical for each for 24 hours. And the endpoint would be turbidity measurement at 405 nanometers as referred to the opacity. Application is it can categorize the chemicals to category 1 and no category. This is a fast, simple, and inexpensive and readily available assay. Similarly, OptiSafe assay is IQVAM validated. Again, it uses the transparent macromolecular matrix. Exposure would be same, but only difference is that it would be a test chemical would be exposed for 18 hours instead of 24 hours. Turbidity measurement would be made again at 405 nanometers for opacity measurement. And this test is used and accepted by US EPA to classify the category number 4 along with UNGHS no category. So this is a representation of a microplate well, wherein this protein matrix and the macromolecular matrix is overlaid with water and test sample and incubated and then tested for its opacity. Coming to the other validated or optimized cytotoxicity methods used for classification of eye damage are neutral red release test and the cytosensor microphysiometer test. Coming to the neutral red release test, it is validated and peer reviewed. And the test system includes confluent monolayer of fibroblast cell line. Exposure would be for 1 to 5 minutes with different concentrations of the test chemical. Endpoint is again using cytotoxicity measurement based on neutral red release from the cells. Application would be the classification possibility to non-category for water-soluble test chemicals, UNGHS category 1 for agrochemical formulations with proof of concept study. That means it is an additional study required for classification. So coming to the cytosensor microphysiometer test, it is again validated and it uses subconfluent monolayer of mouse L929 fibroblast cell line. Exposure is sequential exposure to 7 increasing concentrations, each for 13.5 minutes, followed by washing, and measurement would be for the total exposure would be for 20 minutes of duration. Coming to the endpoint, the cellular metabolic rate will be measured by checking the acidification pH changes using cytosensor microphysiometer apparatus. And this method is used to detect GHS category 1 chemicals only to limited to soluble chemicals and those that form a stable suspension during testing UNGHS and during testing. And when it comes to the UNGHS no category classification, only it is applicable to the soluble surfactants and those that form a stable suspension during testing, and also used by US EPA for categorizing the chemicals as category 3 and 4, which are safe or irritants, non-irritants. So this is a cytosensor microphysiometer. Right now, this meter is not available in the market. So, and the advanced model, when it is available, this method holds applicable for testing. Other organotypic ex vivo assays which are optimized to use as additional non-validated tests for classification under IATA approach, that is integrated approach on testing and assessment, are this is isolated rabbit eye test, porcine cornea rabbit test, and other methods. Coming to the isolated rabbit eye test, the validity is that it is interlaboratory validation is completed and it is peer reviewed. The test system includes ex vivo rabbit eyes mounted on specialized whole globe eye or cornea holders, and it is similar to BCOP assay. The exposure goes for 10 seconds to identify a severe irritant, that is corrosive category 1, and 1 minute or longer for less severe irritating substances. The endpoint is to look in for the corneal swelling and subjectively corneal opacity injury to the epithelium by measurement of fluorescein retention. It is applicable to classify UNGHS category 1, that is the corrosive chemicals, using a top-down approach from IATA. So this is a pictorial representation of the opacity. Then coming to the porcine cornea opacity reversibility assay, PORCORA. It is again an optimized assay which is used as an additional test, non-validated test. And the test system would be excised porcine cornea. These are exposed with test chemicals to 5 minutes. And the endpoint again is the corneal epithelial recovery over 21 days by fluorescein stain retention. Corneal injury reversibility using sodium fluorescein stain or to detect the compromised epithelial barrier function. This assay is used to categorize UNGHS category 1 with all type of chemicals. So this is another pictorial representation. And finally, other in vitro assays include slug mucosal irritation assay, which is again optimized as additional test for classification, uses mucosal surface of slug. It is Arion lusitaniscus. The exposure is made with exposure should be for the slugs as such directly. It is not exposure. The endpoint is the mucus produced from mucosal surface of slugs. LDH activity, protein content are measured using the turbidity at 405, that is the opacity. And the applicability is, this is used for categorization of chemicals as 1, 2A, 2B, and non-category. And the additional test is Hen's egg test, that is a chorioallantoic membrane headcam assay. It is again validated and peer reviewed. The test system includes chorioallantoic membrane cam of fertilized chicken eggs. The exposure would be for 300 seconds, that is almost for 5 minutes. And this assay is used to categorize category 1 based on coagulation and no category based on the coagulation and hemorrhage and lysis of the blood, chicken eye membrane. This is again fast, simple, expensive and inexpensive and readily available method. Additional test is chorioallantoic membrane vascular assay CAMVA, which is again a me-too assay to the head cam. It is validated and blood vessels of the chorioallantoic membrane would be assessed to check for the hemorrhage from fertilized eggs. Exposure is for 30 minutes. Endpoint is vascular change to the cam, such as hemorrhaging or hyperemia, that is the capillary injection, and occurrence of vessels devoid of blood flow, that those are ghost vessels observation. This is applicable to categorize chemicals additionally to category A or B, and this is a pictorial representation. Then coming to the classification and assessment of eye damage, an integrated approach on testing and assessment for serious eye damage and irritation hazard identification is developed in view of replacing the sequential testing strategy as per OECD 405 until 2017. There is a monograph available with OECD which provides guidance on how and when to integrate this existing or newly generated information for decision making, including decisions on the need for further testing or final decisions for classification and labeling of chemicals. So a schematic outline of the IATA for eye hazard identification, focusing and classification and labeling is available in this monograph, which constitutes of 3 parts that guide the assessment of serious eye damage and eye irritation potential hazards. Part 1 has 6 modules where Part 1 guides on existing and non-testing of data. Part 2 guides on weight of evidence approach analysis. And Part 3 guides on the generation of new testing data, where the modules 4, 6, and 3 are used to use OECD adopted in vitro test methods, other non-OECD adopted test methods, and lastly it would be a usage of in vivo animal test, which is exception. And this part 3 includes top-down approach for classification of UNGHS category 1, that is corrosive material, and bottom-up approach for UNGHS no category chemicals. So this is the classification wherein the top-down approach includes in vitro test method to classify it as category 1, and if non-category, go with an additional test and classify it as non-category, and then go with weight of evidence approach to classify the chemical. Bottom-up approach executes an in vitro test method based on OECD adopted method, and in vitro test method additionally for identification of serious eye damage to classify it as non-category. There is an exception for a top-down approach where the collected existing and non-existing information from part 1 is evaluated in this approach, and this top-down approach has an exception that on the it should be used when all available collected informations and the weight of evidence assessment results in a high priori probability that the test chemical may induce serious eye damage. In such cases, this testing with additional tests can be waived off. Then comes to the defined approach methodologies. These methodologies can be used to support the hazard classification, hazard characterization, and or the safety assessment of chemicals as per OECD 467. These methodologies can be used either on its own to reach a conclusion on classification and labeling, or together with other source of informations within IATA. So defined approaches 1, that is DAL1, is based on physicochemical properties of a test chemical and in vitro data retrieved from RHCE and BCOP LLBO methods for neat non-surfactant liquids to classify them under category 1, 2 and non-categories. Defined approaches 2, based on in vitro data, that is directly from BCOP LLBO and short time exposure assays for non-surfactant neat liquids and solids that are dissolved in water soluble. To classify them under UNGH's category 1, 2 and non-category. So there is a graphical representation of predictive capability of these defined approaches where the capability is above 50 and almost up to 85 to 85% with category approach 1 and 2 as well. Recently, LP et al. Have published a paper on usage of defined approaches for surfactants, which was a limitation with other defined approaches. Excuse me. So here the test chemical would be first tested with OECD 492 using validated research method 1 or 2, that is either skin ethic or epiocular models. Please excuse me. And here, if the mean cell viability is greater than 60, then the prediction would be no category. And if it is not, then the measurement would be modified, tested using short-term exposure assay. I'm really sorry, I have
Dr.
some.
Parvathi, take a pause. Take a quite calmly, take some water, take a pause, okay? We'll wait for a minute or two. Don't worry, just feel comfortable first, okay?
Yeah. In the meantime, I'd let all the attendees know that this is the 4th session so far. We've had really good 3 sessions. The first one was by Dr. S.K. Jain, who is the toxicologist at CIBRC, who delivered an excellent presentation on what NAMs can be used to meet pesticide testing requirements. If you were not able to attend that presentation or have not looked at the recording yet, I would highly encourage all the attendees to go back and check out the presentation, which is available at our Science Consortium website. It's very informative and you'll get a very detailed idea of what kind of NAMs can be used for regulatory submissions in India. So.
Yes, I'm okay now. I can continue my session.
Thank you.
All right. Thank you all for sparing me. And when this no prediction is no category prediction is not possible with this method, then modified short time exposure assay is carried out, wherein the mean cell viability, if it is less than 20% at 0.5% concentration, then directly go with the prediction of category 1 classification, and if not, go with the predict category 2. This is a recent proposal proposed by Alapi et al. In 2023. Coming to the decision tree approach, the next step in the tiered strategy is to go with decision tree approach for eye hazard classification, where physicochemical properties are evaluated, followed by testing with available in vitro and ex vivo methods, including BCOP, cytosensor assay, and epiocular, that is RHCE models. These are the primary models which are used for labeling of eye hazard chemicals. This is done based on their respective methods and in vitro scores, wherein the PhysChem properties are first considered. After that, BCOP method is considered based on it in vitro score. The classification to category 1, 2, and 3 are made. If not, then the cytosensor assay is used, where the in vitro MRD score, if it is less than 2 or greater than 80 or in between, can be classified using category US EPA category 4 and 3. And if it is less than 2 mg, to distinguish category 1 from 2, BCOP additional test is used. And coming to the epiocular assays, the in vitro ET50 score, that is using time to toxicity assay. This categorization to category 4, 3, and category 1 and 2 are made. Coming to the label elements for serious eye damage based on these resistant tree approaches and IATA approaches, the UNGHS category carries a symbol of corrosion. UNGHS2 category A carries an exclamatory symbol. Category 2 will not hold any symbol. The signal word is danger for GHS1 category 1, and for 2A it is warning, 2B also it is warning. The hazard statement to be mentioned would be causes serious eye damage for category 1. For category 2, it is causes serious eye irritation. And for category 2B, it is causes eye irritation, which is a mild classification. So this is a brief about all the in vitro and ex vivo assays, which I have listed and discussed about today. And these are few references from which I have made this presentation. And that's it from my side. Thank you. I hope I have not exceeded my time.
No, you were well within your time. Thank you so much for that.
All right. Thank you.
Great.
I
Okay,
would
thank
await the
you so much.
questionnaire session. Yeah.
So we'll be taking questions at the end after the second speaker. So thank you so much once again, Dr. Parvati, for such an informative presentation. Our next speaker today would be Dr. Pradeep Kumar, who would be presenting some case studies using in vitro and ex vivo eye irritation and corrosion test methods. Dr. Pradeep earned his degree in veterinary sciences, Doctor of Veterinary Medicine, Master of Science from Bengaluru Veterinary College. After completing his education, he joined Venus, a preclinical CRO, in 2006 as a study director for general toxicology and safety pharmacology. Dr. Pradeep is a diplomat of the American Board of Toxicology and is currently working with Corteva Agriscience as a regulatory toxicologist. It is now my pleasure to turn the floor over to Dr. Pradeep.
Give me a second, Ankita. Are you able to see my screen?
Yes, sir, it's visible.
Okay. Uh, Good afternoon, everyone. In continuation of the previous talk, I would like to discuss a bit of acute toxicity testing in general, alternate approaches for eye irritation, case study, and a few takeaway messages. Pesticide active ingredient and plant protection products animal testing. Agrochemical active ingredients are one of the most rigorously end-to-end tested group of chemicals on the market. This requirement takes us to the use of 18,000 animals for entire spectrum of testing per active ingredient. Acute toxicity testing is an integral and essential part of testing requirements globally. 50 mammals are used for new product testing from the acute six-pack studies, and one can imagine the number of animals used for testing as hundreds of new products each year across the globe are registered. Key driver for characterizing acute local toxicity properties for the end-use agrochemical products is to provide appropriate hazard communication via labeling for worker and user safety. Plant protection products may have differing acute toxicity potential or compared to active ingredients because of physical properties of plant protection products. For example, granular formation, there may be a possibility of mechanical irritancy. Route of exposure, example, dust formation during a mixing may prone for a dermal, respiratory, or sensitization risk. Toxicity profile of co-formulants. Uh, the formulation will have co-formulants like thinner, binders, solvents, emulsifiers. They will contribute to the overall toxicity of the formulation, and there may be a possibility of additive effects of formulation ingredients that are also a reason for differing acute toxicity potential. Dry test is the primary in vivo method for eye irritation and is accepted by regulatory agencies worldwide. This test is a subjective in vivo test with poor reproducibility, and this dry test was never formally validated for its relevance to humans. There are number of differences between rabbit and human eye. For example, presence of nictitating membrane in rabbits, which essentially sometimes trap or remove the applied test material. pH of rabbit eye aqueous humor is more alkaline than in humans. Rabbits are not efficient as uh, 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 rabbits are not efficient as humans in tear production. So overall, the dries method is often criticized for overestimation of the effects for some aspects of testing from animal welfare reasons and also for reproducibility of the test results. There was a retrospective analysis of in vivo eye irritation test results was done, wherein ICAR database was used for evaluation of substances with 2 or more test results, dries test results. Compounds scoring a category 1, that is eye corrosive, had a 10% chance of having no classification, that is non-irritant, in a subsequent test. And also the reproducibility for category 2A and 2B was worse. For 2A, it was around 60%, and 2B, it was around 80%. The problem is compounded with agrochemical formulations, which are complex mixtures that are often not severely irritating but can have a persistence of effects. Primary eye irritation alternatives. Over the past decades, there have been substantial investments made into the both development of in vitro methods and validation of validation to access the reliability and reproducibility of methods to predict the eye irritation response. Despite these efforts, no standalone in vitro test has successfully been validated to provide or to fully replace the dry irritation test for regulatory purposes. Some of the available alternatives in the in silico prediction field are OECD QSAR toolbox, Derek Nexus, OSAs, and so on. But these tools are not routinely performed for regulatory assessments, and they have low acceptability than in vitro assessments generally. So there are in vitro assessments available. Major ones are OECD 492, 437, and then 438, which I will not go into the details as previous speaker has elaborated them. OECD guidelines and general requirements necessitates evaluation of eye irritation potential before performing in vivo testing. Based on the OECD IATA guidance, we have two approaches for eye irritation testing: top-down and bottom-up approach. There is no single in vitro study that is a replace for in vivo testing requirement. If the test material is predicted to be severe irritant based on existing information like physicochemical properties, then top-down approach is considered and test A is conducted. If test A is positive, then classification is considered as category 1. If it is negative, then test B is conducted, and when with the test B, if the result is negative, then it is considered as non-irritant, and test B results in positive result, then the test material is considered as irritant to eyes. For chemicals or test substance that are predicted to be non-irritant, then bottom-up approach is employed, wherein test B is conducted first, and if the result is negative, then it's considered as non-irritant. If it is positive, then test A is conducted, and if test A is positive, then test material is considered as severe irritant or category 1, and if it is negative, then it's considered as category 2, irritant to eyes. Regulatory frameworks have seen considerable changes in the testing strategy used for hazard characterization of local toxicity in the recent years. In some areas, a complete removal of in vivo testing was implemented as in the cosmetic sector. Agrochemical industry is in a transition stage. At present, the regulatory acceptance is very subjective globally, with options for waiver of in vivo tests. European Union, Australia, New Zealand potentially accepts in vivo tests. In US and Canada, a pilot program is on for future acceptability. In general, Latin American countries and APAC countries still rely on in vivo tests. However, countries like India, Japan, and then Brazil encourages in vitro testing on acute endpoints wherever possible. Some of the advantages of in vitro tests are that they are routinely used as part of active and plant protection products acute toxicity assessments. They help to screen out obviously the corrosive actives formulations before they are ever used in animals. They can be used conservatively for classification following consultation with regulatory agencies, and they have a good negative predictivity. Some of the disadvantages are that they are validated validated in monoconstituent preparations. They tend to have false positive or overproductive potentials, and these in vitro tests have variable acceptance in many geographies. Recently, industry made an effort in collaboration with Crop Life Brazil, wherein retrospective analysis of the eye irritation in vivo and in vitro test data was analyzed. For this project, working group members included agrochemical companies, and the goal was to conduct a retrospective analysis of in vitro eye irritation assay results and GHS additivity calculation for pesticide formulations compared to in vivo GHS regulatory classifications, and to propose testing strategies for evaluation of eye irritation potential. For this project, 8 companies provided data for a total of 187 formulations, and that data included at least one in vivo test, one in vitro test result, and GHS additivity calculation. The out of 187 data points, like 21 formulations were having a GHS category 1, that is 11%. 33 formulations had category 2, and 133 formulations were non-irritants. The performance of each approach was evaluated for concordance, sensitivity, specificity, false positivity or negativity, and negative or positive predictivity. Here is the result of data analysis. I would like to highlight the one outcome for today's purpose, that is negative predictivity. For BCOP, the negative predictivity was 86%, that is 6 out of 7. For ICE, it was 88%, 28 out of 32. For ECHE epiocular, it was 46 out of 46, that's 100%, and calculation method had negative predictivity of 93%, that's 77 out of 83. 4 of these alternative methods more or less correctly identifies the agrochemical formulations, correctly identifies the agrochemical formulations that do not require classification for eye irritation. There is a high confidence that prediction of not classified from alternate method is a true not classified for all 4 approaches. The negative predictivity range is from 86 to 100%. Let's look into an example of a name-based alternate testing approach based on the results. There are 2 steps here. Step 1, which is initial considerations, and step 2 is in vitro eye assessment. In step 1, assess the potential of the formulation or test material for irritation and corrosion. Consider or evaluate reasons for exclusion. These reasons can be like physicochemical properties or historical data, eye corrosion or severe irritation potential, extreme pH, skin corrosivity, which gives a good correlation for either positive or negative effects. Information on co-formulants of concern, or consider any other reasons. So if there are no concerns, then if there are concerns or red flags in this step 1, go back to the drawing board and address the concerns. If there are no concerns, then proceed for step 2, wherein consider the available testing in vitro methods from in silico, in vivo, or ex vivo methods Here, bottom-up approach is considered because severe irritation possibility is already reviewed in the step 1. One or more testing method may be used depending on the test material property and assay relevance and use scenario. If the outcome is negative, then the likely GHS classification is NC. If the result is positive or inconclusive, go back to the step 1 and review the assessment strategies. So overall, there are 3 outcome possibilities. One is NC. Conclusive in vitro results in combination with sound predictive data as a support would make a strong case of in vivo waiver with regulator. Positive or corrosives, revisit the formulation and strategy. If it is inconclusive, again go back to the step 1 and review the data. As a last resort, think of animal testing. With this decision tree approach, animal testing could be avoided in 2 situations out of 3, that is NC or positive results. Some of the takeaway messages of this engagement are that the industry and scientific community are currently in a transitional phase where ag companies are running both in vitro and in vivo tests routinely per OECD guidance. There is a tendency towards over prediction affords greater confidence of human health protection from negative results. That means most of the methods overestimate the irritation potential in humans. High confidence that prediction of not classified from an alternate method is a true not classified for all 4 approaches. The negative predictivity range is 86 to 100%. Given the preponderance of agrochemical formulations that are true negatives, that is 70%, such an approach could significantly reduce the use of animals for this endpoint, which can be up to 50 to 70% of less rabbit tests. Here is the list of some additional references for those interested in further reading. Thank you all.
Okay, so thank you, Dr. Pradeep, for your sharing with us Corteva's work and experience. I am Dr. Akbar Shah. I am the General Secretary of the Society for Alternatives to Animal Experiments. And before that, I have been the Professor of Animal Science at Bharatdasan University, Tiruchirappalli. And later I established the Center for Alternatives, the first center in India. So it has been a long experience, involvement in alternatives movement in India. So with this brief introduction about me, I am requested to be moderating the questions and answer period. We have, in addition to the 2 speakers, Dr. S.K. Jain, the Senior Consultant at Toxicology from the Central Insecticides Board and Registration Committee, and he has joined us as a panelist today. So we have, we can have some questions from the audience. So if there are questions, the audience can bring up the questions and can be addressed to the speakers directly if you have. Is there anybody trying to raise questions or bring up questions to the speakers? They can do it personally straight to the speakers. In Kida, is there a possibility somebody will be asking questions, putting questions directly to the speakers?
I can take up the questions, if that's okay.
Yeah, please.
Okay. I see the first question for Dr. Parvati here. Dr. Parvati, can you elaborate a little bit on the difference between the top-down and the bottom-up approach?
Yes, Ankita. So the top-down approach is used to categorize the chemicals as category 1, and bottom-up approach is meant for categorizing the no category, that is the non-corrosive or non-irritant chemicals. So when it comes to the top-down approach, there is a requirement for additional test along with a single uh, in vitro alternative method. It means that a single method is not sufficient enough to adjudge or justify the categorization. So an additional test will be conducted, wherein the defined approaches, if you could see from my slides, there are two approaches where initially BCOP assay is used and followed by short time exposure assay or RHCE. And when it comes to the other defined approach too, it directly goes with RHCE and STE kind of testing. So this is how the trend goes with top down. And there is an exception, as I told you, with this assay, that if the existing data and the historical or the weight of evidence approach shows that the chemical is clearly a corrosive kind of thing, then it can be easily, the additional test can be waived off from the top-down approach. So I think I have answered you.
Thank you so
And
much,
my
Dr. Parvati.
co-speaker would be the right person because he's into regulatory, so he would touch upon that elaboratively.
Okay. Could you let us know for BCOP assay, what would be the positive control that is used?
Yes, I can. Just a minute. It is methyl acetate usually which is used.
Okay. Next question is for Dr. Pradeep. Dr. Pradeep, you showed a very interesting slide about the regulatory acceptance of such methods in different countries. And we see that there is a lack of international harmonization. Why do you think, this is the scenario, and how do you think this situation can be addressed? Because there are so many methods which have great acceptability in certain regions, but may not have been accepted by other regulators. So what could be the reason, and how can this situation be addressed? Dr. Pradeep, we can't hear you.
Maybe he has to unmute the mic.
Are you able to hear me now,
Yes.
Yeah.
Ankita?
Yes, sir.
Yes, can hear you.
So this is a evolving situation. Efforts are underway to refine the available in vitro methods for the desired outcome, and also for the methods or new methods are being validated to address the gaps. Hopefully, in the coming years, we would see full replacement methods for animal testing, and essentially that will bring the harmonization of guidelines across the globe, hopefully. Having said that, I would like to listen from SK Jain on this question.
Dr. Jain, we can't hear you. Dr. Jain, you'll have to unmute yourself. I think that should be fine.
Okay.
Yeah.
Thank you, Dr. Pradeep. We have gone through in detail regarding the in vitro methods for acute, particularly the oral testing or the eye testing. And we have found that these tests can be tried. At least before we go for the animal testing, we can try the in vitro test or ex vivo test and see the preliminary endpoints, that what are the basic results, what they indicate, and whether there is any requirement for further animal testing or no. We can rule out in the beginning. If the product is very, very safe, and say in eyes it's not irritant at all, then we can rule out, and I hope various countries should accept these non-alternate animal methods where we can avoid use of animals or minimize use of animals. And we are trying, and we are trying to revise our guidelines and give a lot of options for the applicants, registrants, to file their applications where non-animal methods can be used and the data can be submitted, which can be evaluated and if found proper, can be allowed without animal testing. So India, I think, is one of the country which is quite going forward in this direction.
Dr. Ankita, can I respond a little bit? Just add upon something here? Okay, yeah, sure. Thanks, Dr. Pradeep and Dr. Jan. Dr. Pradeep, I'm not sure whether you're aware of, but during when this 2017 guidance harmonized document came across after approximately 2 years of industry, deliberations along with PETA and Humane Society International. And at that point of time, I was the executive director of CropLife India. So it was during those deliberation, it was agreed upon it, and it has been minuted that CropLife would have given at least in vivo data vis-a-vis alternate animal data to the regulators on 10 molecules, which I'm not sure whether that has been adhered to at the moment or not. And therefore, then questioning the regulator itself that by when can we expect them to, start accepting in vitro methodologies may be a challenge. I would, at this point of time, just like to suggest you that if you could ask your colleagues at CropLife, they may be knowing about it, those who are, into the deliberations, that this thing needs to be done, and CropLife as such can provide both methodologies, in vivo as well as in vitro, for a comparison to be made by the regulator to have a confidence to take future acceptability towards in vitro testing without any in vivo studies in that regard. So I believe it will be more relevant that earlier decisions which have been forgotten can be taken across for a speedy acceptance of in vitro or non animal studies. Okay, that's just all. Just wanted to give some feedback on that part.
Good to know, Dr. Saini.
Thank you, Mr. Saini.
Mr. Saini, you are right. I think CLI has not taken any initiative after that, and probably they will be taking soon. And as soon as we get the data, comparative data, and go through, and then we can go ahead and start doing the applications without animal testing.
Exactly, sir. And this decision was taken way back in 2015. I left in 2016. So yeah, it's been quite some time. So probably Dr. Pradeep Kumar can take it over again, and let's hope something goes on fast on that part.
We hope so.
We'll look into it. Thank you very much. Thank you.
So moving on to the next question, Dr. Pradeep, is again for you. So you demonstrated how there was a high negative predictability with all the 4 methods that you showed here. In case there is a positive result seen in these in vitro methods, does that necessarily trigger additional animal testing? And if yes, is there a way, to avoid animal testing using other strategies, or what other, options are there if we do not want to conduct additional in vitro testing?
Yeah, there are two possibilities here. One is internally at the company level, if there are red flags or positives, it's always better to look back and look into the prediction models, do additional tests, or revisit the formulation ingredients for toxicity, and refine the formulation. That's always a possibility, within the company before getting into the in vivo testing. The other one is the regulatory scenario. Probably we need to go with the regulatory acceptance from the different geographies as well.
Okay. So, Parvati, ma'am, under what circumstances could waivers be considered for eye testing?
Can you repeat it, Ankita?
Under what circumstances can waivers be submitted for eye testing?
Waivers you are saying about?
Yeah.
For eye testing. Okay. When there is a strong data available from the literature and the historical data, which shows that the predictive capacity would be greater than 80%. Maybe my co-speaker can correct me if it is. So I believe that eye testing can be waived. Dr. Pradeep, can you emphasize on this?
Yeah, probably a clear conclusive result on the negative side would be an interest for regulators. But again, I will pass this on to SK Jain for his comments.
Yeah, you are right. I think it depends on the type of physicochemical properties. When the compound is well known that it is not at all corrosive, and the formulations which contain different impurities, they are also known to be non-corrosive, I think it will be more than 80 or even 90% chances that the formulation ultimately must be corrosive, and in those circumstances can be waived off, can be considered by the regulator. And if such case comes to me, certainly I will review and accept it.
Thank you, Dr. Jain, for clarifying that. I think that's really helpful for the registrants to know. Dr. Parvati, a very general question: are these alternative methods available to CROs in India? And out of the alternative methods you talked about, what all are being offered by Aurigene?
All right. So we at Aurigene are, providing services to pharma at present with alternative methods like in vitro skin irritation and corrosion models, eye irritation corrosion models, and skin sensitization models, wherein the key events 1 and 2 are addressed. Yes.
I
so
see. Okay.
Other assays are under validation. So soon we will put up them into our portfolio.
Okay. Thank you, Dr. Parvati.
Thank you.
I see one last question for Dr. Pradeep. Dr. Pradeep, you talked about the GHS calculation approach. Can you expand a little bit on that, and whether such approaches are, which countries are accepting these approaches globally? If you could share a little bit of insight on that.
So GHS additivity approach is a computational method with which UN GHS classification system based on theory of additivity. So it predicts mixture toxicity without experimentation. So the formula uses toxicity information of all components of mixture for prediction of overall toxicity or classification.
And what about the regulatory acceptance of this approach? I mean,
yeah, on a case-by-case basis, like we have experience of submitting them to the regulators, and we have seen the positive results as well.
okay. Again, helpful to know. Thank you so much. That would be all the questions from the audience. Dr. Jain, do you have any questions in particular for the speakers?
I want to know that whether it is acceptable in North America or in other Latin American countries, the mixture formulations and the mixture toxicity. Because we are also thinking to implement in India also that without going into the detailed toxicological profiles, if it can be allowed, can be considered.
Dr. Jain, I don't have that information available immediately with me, but I will gather that information and pass it on to you later.
Yeah, that will be helpful. Thank you.
Thank you.
Any question for Dr. Parvathi, Dr. Jain?
No, I think it was a very excellent presentation by both Dr. Parvati and Dr. Pradeep, and I hope it will be quite useful to all the participants and all other registrants.
Thank you.
Thank you.
100%.
So I think I
Dr.
should take over now. Yeah. So it's my turn to record our appreciation to the wonderful presentations by Dr. Parvati and Dr. Pradeep Kumar. We really did enjoy the presentations, and we are enlightened of the alternative test methods which are in the offering to be used. And I think the participants who joined this webinar are really enriched with knowledge on the alternative methods. And I thank the people who joined this webinar as participants, and they made it. We are motivated to be in service to you in the days to come. And if the attendees have questions or comments, please write to Dr. Ankita Pandey. And as the webinar ends, there will be a pop-up to answer a few survey questions on today's session. Please do spare a few minutes to answer these, and we will appreciate your feedback. The next session is scheduled on the use of QSARs for the pesticide toxicity assessment. That will be on 7th July. So once again, thanking you for your wonderful presentations and your participation. And I wish you to enjoy the rest of the day. See you next time on July 7th. Thank you very much. And thank you, Ankita. Thank you, Dr. Jain. We should thank Dr. Jain especially. I forgot to say that. It's really enriched.
We still have 10 minutes. I'm really sorry. I was wondering if Mr. Saini, I think he should have an opportunity to ask any questions if he has. So in 5 more minutes, we still have 10 minutes of our time here. So Mr. Saini, please go ahead and ask any questions.
Thanks, Ankita, and thanks, Dr. Akbar, for concluding the session anyway. So a lovely presentation as usual by the presenters, and I have just got 2 set of questions. One is directly to Dr. Parvati. Dr. Parvati, I noticed that you said that the corneas, bovine corneas are bovine eyes are taken across from slaughterhouses. I would like to know whether there's any specific procedure to this part, and when and how do you acclimatize those isolated parts as an alternate method?
Yes,
Just curious on that part. Yeah.
All right. If I understood your question correctly, it is that the management of the bovine eyes immediately after the slaughtering and collection of the eyes to the test conduct exposure time. That is how I am understanding your question.
No, but
So
is there an SOP to that? Is there a procedure?
yes, there is a TB-ALM protocol available for BCOP test method. It is clearly, elaborated. And initially, we'll have a tie-up with a slaughterhouse in our city. And even before we plan the activity, we inform them that these many eyes are required for our testing. And on the same day, we approach them, and there will be a person to collect those eyes, and we bring it immediately to the lab, and we select the healthy cornea, and we will reject most of the corneas because they will be either opaque or damaged, or they'll have some kind of a defect. So it is kind of that from the slaughterhouse it will be a huge collection of cornea, the eyes of a bovine, and we select the better and testable cornea, and then we separate the cornea from the bovine eyes and place them onto the holders. So it happens that we plan the experiment on a single day, wherein the collection and selection, treatment, and analysis goes on the same day.
On the same day. Yeah, fine. Thank you very much. If you could be kind enough to share the protocol number or the protocol itself.
Yes, I would do that.
I believe it would be nice for all viewers to have a collection of that.
Definitely.
My second question is, thank you very much, Dr. Parvathi. My second question is basically directed to both of you, but primarily on the comment given by Dr. Kumar. Dr. Kumar, in your presentation, you stated across that no single in vitro test is there for replacing an in vivo test. Is my understanding right? I mean, in the current scenario, whatever animal testing we have, there is not a single in vitro or an alternate animal methodology that can be placed in to replace an in vivo method. Is my understanding correct on this regard, Doctor?
Yes.
Okay, so my question to both of you is then, by when do you expect that we can have a one-to-one solution, one alternate animal methodology that can replace a total in vivo test in that regard? I mean, how do you foresee the future? I know it's a tough one, probably, but
yeah, I don't have an answer.
Have an insight on what's happening across in the globe on that front.
All right. I don't have an answer, but it may not be like one-on-one, but a combination of in vitro effects, probably a reality soon.
Okay.
Yes, to add upon Dr. Saini, it is that the sensitivity and specificity of the assay, which accounts to the consideration of a single test for this classification of chemicals. So it is well articulated in the monograph, which I will share with Ankita, and she must be having that as well. So it is mandated that an additional test is required, to my understanding. Correct me if I'm wrong, Dr. Pradeep.
No, I believe we got the answer. I mean, it's just out of curiosity that, there is a lot of rumors that go around that in vivo is probably a cheaper version than alternate methodologies. But then again, I do agree and fully support that alternate animal methodologies need to be, taken across more seriously. So, I mean, I was just wondering if something can come across, but maybe in our subsequent episodes, and so probably we'll have some kind of clarity on that, or over the past, in the distant near future. Okay. Thank you very much, both of you. Thank you.
Mr. Sani, I just curious about one thing. So we understand we may not have like a standalone replacement for the in vivo test currently, but when these different methods they used in a particular combination, is there a way that covers, I mean, is there a combination of approaches that could cover the entire span of the classification scheme?
Not like we have one test and or two tests that determines or covers the whole classification. As of now, like there are different aspects we need to consider. That is the test material property and also the application where we use the material. So application scenario also we need to consider. And moreover, like we have different in vitro methods, their advantages and disadvantages had to be looked into. And then that need to be matched with the test material properties. So with that, we will get the desired outcome. So there is nothing like do one and then two and then get the result. So that's the situation as of now.
Thank you. And I completely agree with Mr. Saini. I think cost is definitely a limitation with the use of in vitro methods, but just like the last speaker mentioned that, the more and more regulatory acceptance that these in vitro methods have, I think the cost is definitely, bound to go down. So
yeah, Ankita, that's what I'm trying to understand, that, in the distant near future, at least we can hope that something comes across with enough data in the archival pool, which can be summarized depending upon the physical chemical properties of various molecules coming across and all that kind of stuff. So yeah, it's really an insight into that whole issue. Thank you once again, all the presenters, and Dr. Jan and Dr. Akbar Shah for accommodating. Thank you very much.
thank you.
So thank you, and then we'll call it a day.
Thank you all for your time. Thanks a lot.
Yes, thank you all for allowing us to present and share our knowledge here with all the peers like Dr. S.K. Jain, Vipin Saini, and Dr. Akbar Shah. It was really, a great moment for me to present my views onto this eye irritation models because I have been working on in vitro toxicological studies where alternative methods have been my primary area of research, along with genetic toxicology, because I started my career as a genetic toxicologist. So I foresee that there would be a lot of scope for in chemico, in silico, and in vitro models to get accounted for classification of chemicals. So I really look forward for the next presentation, and thank you so much for giving this opportunity.
Thank you.
Thank you.
And Dr. Pradeep, it was a very nice presentation from your end, and I appreciate that.
Thank you, Dr. Parvathi.
Thank you all.
Okay,
Thank
bye.
you.
Bye.
Have a good evening.
Bye.
Good evening.
Thank you.
Bye.
Okay, thank you very, very much, everybody.
Okay, thank you.
Thank you, Ankita. See you again.